Hello, everyone. Welcome to our biological mystery case. We have a hypothetical case with a patient who's feeling sick. And with winter time already coming up, we aren't sure if this is caused due to a bacteria. And the way we'll solve this case is to identify the bacteria and identify if this is a bacterial infection. In order to solve the case, we need to first find a way to check for the bacteria. Oh, wait, there's a laboratory technique and it's called gram staining. But first, what is a bacteria? It's a unicellular organism that can live in many places in the body, with some causing infections and disease. These gram staining tests can help to diagnose infections and detect the type of bacteria. The gram staining technique was developed in 1884 by Hans Christian Gram, a Danish microbiologist who classified these bacteria into two distinct groups, gram positive and gram negative.
Here we have the gram staining procedure. There are two cultures, yellow and green, that we want to investigate using a gram stain. The first step of gram staining involves preparing a fixed slide. Inoculation involves using a loop to transfer a drop from the sample culture to a fresh microscope slide. Next, smearing involves spreading the culture evenly onto the slide into a thin film. Lastly, the slide is moved over a Bunsen burner briefly to heat and fix the sample on the slide. Now the slides are both prepared, our next step involves staining the slides. First, crystal violet is placed over the fixed slides and then rinsed off carefully after 15 seconds. Next, an iodine solution is added to the slides to fix the dye and then rinsed off like before after 15 seconds. After that, a decolorizer, which removes the dye, is added onto the slides for 5 seconds before being rinsed off with water. Lastly, a counter stain called fusion is placed on the slides for 15 seconds before being washed off with water. Now that the gram staining is over, we can say that the yellow colony has gram positive bacteria and the green colony has gram negative bacteria.
The gram stain changes the color of the bacteria, and if it changes to either red or pink, it is called gram negative, and if it remains purple, it is called gram positive. Diving into the clinical applications, we can see some clear differences in positive and negative gram stains. Gram positive infections include strep infections and Staphylococcus aureus. Gram negative infections include salmonella, pneumonia, and urinary tract infections. Looking at the patient sample here, we can see that the bacteria turned purple, thus indicating that this is a gram positive bacteria. Gram positive bacteria are those with cell walls that contain 90% of these thick layers of peptidoglycan. Peptidoglycan is a substance that forms the cell walls of these bacteria. The difference between gram positive and negative is that gram negative bacteria contain very thin layers of peptidoglycan with an outer layer of lipopolysaccharide, which are molecules containing fats and sugars. Gram staining techniques are helpful for healthcare professionals to diagnose and treat infections caused by bacteria, such as UTIs, which are urinary tract infections, and bacterial pneumonia. The most common gram stain tests include wound samples by taking a swab from the site of the wound, blood tests, urine tests, or a throat culture, which can be collected from the bacterial infection like strep throat, taking a swab from your throat and tonsils in your mouth.
Next, we are going to be looking at some of the mistakes that can happen when preparing a gram stain. It is important to remember that gram staining requires practice and is an art just as much as a science. The first common mistake that can occur when completing a gram stain is that you don't see anything on your slides. This is probably because the smear was not properly heat fixed and the bacteria may have washed off the slide during staining. Heat fixing is essentially the process of passing the slide with your specimen under a heat source a couple of times very quickly. The next common mistake that you can see is that all of the cells on your slide are purple. This is probably because the decolorization step was too short, and next time, increasing the duration for this step can lead to more successful results. Decolorization is essentially when the sample on the slide is rinsed with acetone or alcohol for 1 to 5 seconds and then is rinsed with water. Finally, another mistake that is often seen is that all the cells appear to be pink. This likely means that the decolorization step was too long, and next time decreasing the duration for this step will lead to more successful results.
Now that we've looked into common mistakes of gram staining, let's look at how to troubleshoot these. When first starting with the gram staining, it's important that we prepare the stain properly. Bacteria are microscopic single-celled organisms. Therefore, it's unnecessary to make a very thick smear. In fact, this will simply make it harder to decolorize the bacteria. The amount of inoculum that you pick up from a solid medium should be the size of a pinpoint. Additionally, if you're using bacteria from a solid medium, it's important to put saline or water before or after adding the bacteria. Before adding the next reagent, you must wash the slide with water. Once you've done this, be sure to tilt the slide and blot off any excess water. Leaving a bubble of water may cause the reagent to not reach the bacteria or be heavily diluted.